Glycolysis. Glycolysis means sugar splitting, which is the chemical breakdown of glucose. Glycolysis happens outside of the mitochondria. There are two phases in glycolysis, which are energy investment phase and energy payoff phase. In energy investment phase, ATP is used. In energy payoff phase, ATP is produced. It is as simple as that. There are 10 enzyme catalyzed chemical reactions, 5 for each phases. Let's start. In the energy investment phase, ATP is used to oxidize glucose by giving off phosphate groups to each end of the 6-carbon molecule. Glucose is then split into 2 3-carbon molecules, and they are known as glyceraldehyde 3-phosphate or G3P. In the energy payoff phase, 2 NAD+ are used to oxidize glyceraldehyde 3-phosphate by picking up electrons and hydrogen ions, forming 2 NADH. The oxidation of G3P is exergonic, which means that the energy released are used to attach phosphate groups to the 2 3-carbon molecules. Then, the phosphate groups are transferred to ADP, forming 2 ATP, which pay off the 2 ATP used in the first phase. This process of producing ATP is known as oxidative phosphorylation. More chemical reactions occur, changing the structure of the molecules. Finally, more phosphate groups are transferred to ADP, making more ATP. The final products are known as pyruvates, which will be oxidized into acetyl-CoA before entering the citric acid cycle.
Assalamualaikum dan hai, saya Faiz. Saya akan sambung penerangan tentang pyruvate oxidation. Molekul pyruvate bergerak dari cytosol ke mitochondrion. CO2 dibebaskan, NAD+ diturunkan kepada NADH. Coenzim A dan NAD+ diperlukan untuk menghasilkan acetyl coenzim A. Secara ringkasnya, pyruvate menjalani proses oksidatif decarbosylation untuk menghasilkan acetyl coenzim A dan hasil ialah seperti yang berikut.
You begin with 1 glucose and end with 2 pyruvate. It's
Okey, tu teaser je. Tak muat nak letak dalam video, jadi link akan diberi kemudian.
Krebs cycle, or in another name, citric acid cycle, is the continuation of the aerobic respiration. In the first step, acetyl coenzyme A will combine with oxaloacetate to form citrate or citric acid. A molecule of water will combine with the coenzyme A from acetyl coenzyme A to form HS coenzyme A. In the second step, citrate will become isocitrate by changing its molecular shape. In the third step, isocitrate will release 1 molecule of carbon dioxide to form alpha-ketoglutarate, and in the process, 1 molecule of NAD+ will become NADH. Alpha-ketoglutarate will then release 1 molecule of carbon dioxide, and with the addition of 1 molecule of coenzyme A to form succinyl coenzyme A, and in the process another NAD+ becomes NADH. Succinyl coenzyme A will then release its coenzyme A to form succinate. In the process, 1 phosphate will go to GDP to form GTP. Next, fumarate will then combine with a molecule of water to form malate. Succinate will then become fumarate, and 1 FAD molecule will become FADH2 molecule. Finally, malate will become oxaloacetate and a molecule of NADH will be formed. The process will then be repeated.
Our next topic adalah oxidative phosphorylation. Ia adalah gabungan electron transport change dan chemiosmosis. So, in this section, you will learn first how the electron transport chain works and then how electron flow down the chain is coupled to ATP synthesis. So now, let's take a closer look to the electron transport chain. This is the diagram of ETC. Semasa ETC, electron carrier akan mengalami penurunan dan pengoksidaan kerana mereka menerima lalu menderma elektron. Setiap komponen akan mengalami penurunan apabila dia menerima elektron daripada jiran atas dia, lalu dia menjadi lebih elektronegatif sebab dia ada elektron. Lepas tu dia mengalami pengoksidaan sebab dia pas elektron kat jiran bawah dia pula. Lepas tu jiran bawah dia pun jadilah lebih elektronegatif daripada dia. Untuk lebih detail lagi, I will describe the passage of electron through complex 1. Elektron yang terhasil semasa glycolysis dan citric acid cycle ditransferkan daripada NADH kepada first molecule kita iaitu FMN. Lepas tu FMN pun mengalami pengoksidaan sebab dipas elektron dekat ion sulfur protein FeS. Lepas tu ion sulfur protein tu pun pas elektron dekat coenzyme Q ataupun kita panggil dia COQ. Lepas tu dekat kompleks 2 pula, another source of electron for the ETC is FADH2, the other reduced product of the citric acid cycle. Okey, kita sambung balik dekat Q. Lepas Q tu mostly adalah protein bernama cytochromes. Dalam ETC ni dia ada banyak cytochromes. So kita namakan CYT ditambah dengan huruf dan nombor untuk cakap yang diorang ni adalah protein yang berbeza. Cytochrome paling last, CYTA3 akan pas elektron dekat oksigen yang sangat elektronegatif. Dan setiap oksigen akan bergabung dengan sepasang ion hidrogen daripada larutan aqueous lalu membentuk air.
Hi, I'm Fatiha. I'm going to talk a little bit about chemiosmosis. So, what is chemiosmosis actually? It is an energy coupling mechanism that uses energy stored in the form of a hydrogen ion gradient across a membrane to drive cellular work such as the synthesis of ATP. ATP synthase is a complex of several membrane proteins that function in chemiosmosis with adjacent electron transport chains using energy of a hydrogen ion, which is a proton concentration gradient, to make ATP. ATP synthase are found in the inner mitochondrial membranes of eukaryotic cells and in the plasma membranes of prokaryotes. So let's see, this is the mitochondrial structure. As you can see, the white circle there is where the process chemiosmosis occurs. So let's watch this video.
Number of hydrogen ions on the two sides of the membrane. This difference is a hydrogen ion or proton concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP.
Alright guys, so that's all from me. Thank you.
Hello everyone. Let's learn about anaerobic respiration by me, Nur Adina. So, anaerobic secara umumnya ialah tentang the absence of oxygen. There are 2 types of anaerob, which is the first one is facultative anaerob. It tolerates the presence of oxygen. Example of it is bacteria E. coli in food preservation. Facultative anaerob are bacteria that can grow in both presence or absence of oxygen. Another one is obligate anaerobe. Obligate anaerobes cannot live in presence of oxygen, and organisms such as bacterium that can live only in the absence of oxygen. Obligate anaerobes metabolize energy by anaerobic respiration or fermentation. So, kesimpulan yang saya boleh buat ialah obligate anaerobe ni, dia kalau ada oksigen, dia tak boleh digunakan. Dia memang kena tak ada. Tapi kalau figuratif, kalau ada oksigen, dia guna oksigen. Kalau tak ada oksigen, dia boleh guna fermentation. So, I think that's all for me. Thank you.
Hi, I'm Amira and I will be talking about fermentation. Okay, fermentation ni dia anaerobic respiration. So, fermentation ni dia tak perlukan oksigen pun untuk hasilkan ATP. Fermentation can be divided into two, lactic acid fermentation and alcohol fermentation. Lactic acid fermentation happen in human muscle cell, while alcohol fermentation happen in yeast. These two fermentation process will occur after glycolysis.
Okay, before I start into alcohol fermentation, remember that in these two fermentation process, it will begin with the pyruvate, which is the end product of the glycolysis process. Okay, for alcohol fermentation, the pyruvate will undergo decarboxylation and release carbon dioxide to form acetaldehyde. Acetaldehyde ni dia akan direduce oleh NADH untuk menghasilkan ethanol. So, setiap pyruvate ni dia akan produce 1 ethanol, 1 ATP and 1 carbon dioxide. There is 2 pyruvate, so the end product of the alcohol fermentation will be 2 ethanol, 2 ATP and 2 carbon dioxide.
Okay, next kita cakap pasal lactic acid fermentation. Lactic acid fermentation ni dia quite simple compared to alcohol fermentation sebab the pyruvate will be directly reduced by NADH to produce lactate, which means there is no intermediate in this process. So the end product for lactic acid fermentation will be 2 ATP and 2 lactate. Okay, lastly, the accumulation of lactic acid in the muscle will cause muscle cramp to happen. Okay, that's all from us. Thank you.